Oh no. Oh bother. Okay, guys. Hi. My name is Greg. You know what's funny is that like ever since I started doing science communication, doing presentations have been so different. I've really been able to kind of like talk out of my just babble. You know what I mean? Like think about this. This is an hour long presentation, and I told my wife, and she was like, "Oh my god, how are you going to be able to talk that long about this?" And I was like, Kristen, I do an hour, I do like 4 or 5 hours a night. I'm like, are you kidding me? Okay, okay, Bailey. All right, all right. That's a dad joke, guys. Now that is a dad joke. Okay, that was a dad joke. Okay, I know, fishy. Okay, so guys, with peace and love, I'm not going to be able to look at chat. I'm not going to be able to look at that. Okay, thank you, Makari. Thank you. Yeah, I am probably going to wear this. I'm going to wear a different jacket. I'm going to do my green jacket, and I'm going to wear this boutonniere though. Yeah, Helen, this is a cool episode. I mean, honestly, this is really different, guys. This is a presentation for who I am as a scientist, what I've done, and where I kind of see this job going. So, this is a really unique thing. I've actually been looking forward to doing this. I haven't done this before, outlining what I did in my PhD, what I've done before scientifically. I'm highlighting specifically what would be good for this job. So I am talking about my host strain development. Hey, Kathleen, welcome in. This is a special episode of the Dr. Greg Show. This isn't actually, you know what? This isn't an episode of Dr. Greg. This is like a special report. It's like a Dr. Greg special report. Yeah, that's a good one. That's what I'll do that for. So we'll clip this and post it as a video on YouTube. And but you guys can like still chat. I still would love for you guys to stay here, ask questions. Um, so then see what kind of scientist I am. Or if I'm just making it all up. What if I'm just making it all up? Maybe I am. Maybe I am, guys. What if I'm just selling boxes of ivermectin? Nope. No, just kidding. No, it's just kidding. Okay. All right. Okay. Actually, I should get some water though. I am AI. This is true. I am AI. Thank you. Thank you, Helen, for outing me. Um. Okay. All right, guys. Well, let's start. Okay. Here we go. I do kind of want some water. Okay. I'll just have to do some coffee. Look at this. Look at this Sotomayor mug, dude. How freaking sick is it? She's definitely my favorite SCOTUS justice right now. She's so cool. Freaking love Sotomayor. Okay. Yeah. Sotomayor's awesome. Yeah, she is really cool. Okay. All right, guys, for real. Thank you, chat. I'm gonna minimize chat. I'm not gonna look at things, um, and I'm just gonna try to do this. So, thank you guys for being here. Thank you for watching. I'm just gonna minimize chat and and really actually focus on this. Okay. All right. So I'm just gonna act as if I'm talking to myself here. All right. Oh my gosh, everything's all gone. It really does feel weird. Is this thing on? Hello. Hello, world. Hello, world. All right. 35 after. All right. Okay. Here. Here we go. So let me just make sure I'm this. Okay. So Hello, world. I'm just kidding. All right. I was just making sure everything's okay. All right, guys. See you on the other side. 1 hour. All right. Well, thank you for coming, everyone. My name is Greg Newkirk, and I'm just going to go through my research background and show uh, the talents and the research that I've done that I think that can help inform this position and this company in order to make it a better place. So, my focus as a scientist has always been on renewable and sustainable solutions for humans and developing and researching new technologies around that. So, I've worked with land plants and been a molecular biology researcher there, doing next generation sequencing and qPCR screening of mutants, and also programming liquid handlers in order to do genomic DNA extractions, PCR preps, next generation sequencing library preps. In addition, I've also done genome engineering, microbial genome engineering of Eurobia, and I've led a team of that's developed CRISPR there. And I've also worked with Pichia, so developing high throughput strain engineering solutions there. In addition, I have a microbial research background with a PhD where I was looking at solutions and technology in order to enable molecular biology, researching things about the cell that would impact how they took up nanoparticles, and looked into chemical engineering aspects of creating these nanotechnology solutions. And basically, I've been able to acquire those research and development skills through my years of working, 16 years in the lab now, between my undergrad, which was at an algae biofuels lab at UCSD with the Mayfield lab. I've worked at Cebus, which is a plant biotechnology company, and at their automation teams, their host development teams, and their NGS teams. In addition, at UCR, I did my PhD research in the Geraldo lab, where we were doing the nanotechnology-based solutions for plants and algae. And then finally, at BASF Enzymes, where I was working on yeast host strain development and creating those strains that would enable that research. So after undergrad and after I graduated, I was really interested in molecular biology and utilizing molecular biology for renewable and sustainable solutions. So I wanted to go to a plant biotechnology company. Cebus also has a sister company, Nucellus, that does yeast work. And because of my molecular biology background, I was able to work on their Euroia host strain development, where we focused on lipid production. So in this role, I was a strain engineer. And basically what I would do is I would have to find genome targets and I would have to knock them out, and did all the design and molecular biology, knocking them out, and then the final confirmation before handing it off to the fermentation team. Then basically the batch would be ran, and then I would have to coordinate this large scale production in order to get samples. And with these samples, then I would do analyses. So I was doing mRNA expression analysis and sequencing on the batches that came back, confirming the genome engineering hypotheses that we would have. So we would be, working on different pathways of creating lipids, and I was in charge of creating those strains and then analyzing those strains once they come back, with coordination between those teams. And this resulted in a really nice thing for the companies, and we were able to increase by about tenfold the lipid that we wanted to produce, and that was before we had a huge major breakthrough. So in 2013, while doing host strain development, there was the seminal paper, 2013 DiCarlo et al., the Saccharomyces cerevisiae paper with the Zhang and Church lab. Obviously, it was a huge moment for yeast strain engineering, and I basically spearheaded the use and creation of the CRISPR tools in-house, when there wasn't any published research there. So it was kind of us using Saccharomyces cerevisiae papers and other papers that were published in that area to, for the first time, create that tool inside of Euroia for the first time, which is a really cool a moment in my research background, because I was really kind of on that cutting edge where I was really applying publications to solve problems that we had in the lab. And it was a very, very, very fun time. And one of the reasons why I wanted to go get my PhD afterwards, because I wanted that feeling of being on that cutting edge and seeing where the research and development went. Also, at CBIS, I was a part of the liquid handler automation team and the next generation sequencing team. So I eventually programmed like the entire NGS library prep into Hamilton Star liquid handlers, where we were able to high throughput create these NGS libraries. And also being on the NGS team, I was responsible for designing, testing, deploying these, so actually creating the library, and then sending it out to Illumina, and then getting those reads back where we would analyze and develop the workflow, and then send it off to the other teams to give an analysis of how their CRISPR knockout targets did. So we would look at analyses and send it back all within a week. So and after this, it was a really fun experience, but I really wanted to push myself in order to get my PhD. And my PhD was in nanotechnology approaches for Arabidopsis and Chlamydomonas chloroplast bioengineering. And I did it at UCR in the Geraldo lab. And I was lucky enough to receive the National Defense Science and Engineering Graduate Fellowship to fund this research. So basically, the starting point and why nanotechnology approaches are so good it's because microbes are really the final frontier. So 99%, over 99% of microbes are uncultured or unsequenced. So what that means is that we haven't been able to grow them in the lab, or we haven't been able to sequence their genomes. And they have a breadth of abilities that are native to them. So algae has renewable fuel possibilities. In addition, they could actually, algae and other microbes could be used to create biodegradable materials. So, and then on top of that, they could also be used for food. So algae could be used for, protein production, things like that. So, microbiology is really fundamental, and fermentation is really the bedrock for, in my opinion, the future of fuels, materials, and food production. And basically, in order to facilitate this and how we kind of work with the chloroplast, which is fundamental to algae biotechnology, is the chloroplast, but the DNA delivery methods are stagnant. Basically, the current SOP is a gene gun particle bombardment, which doesn't have any organelle specificity. So it's just a broad shotgun style approach where these particles get into, shear the cells and get into these tissues, but they have no ability to be modified. So you can't coat them with anything specifically, and the rates are extremely low. It's a really low efficiency, and then on top of that, basically only about 10 species have chloroplast transformation. So out of all the different land plants, all the different algae, very, very low transformation abilities out of all the plants. And that really limits us to the amount of products that we can make and the hosts that we can make them in. So the chloroplast transformation rates specifically are also very limiting. So basically, the photosynthetic pathways that would be needed in order to study a protein like rubisco, which is the most abundant protein in the world, in order to facilitate mutant library generation of rubiscos, you would need a lot higher magnitude. So this is the complex bacteria-based photosynthetic pathway that had to be put into bacteria in order to actually facilitate rubisco protein directed protein evolution. And unfortunately, this one didn't yield anything, but because there was really only 80,000 mutants that were able to be screened of Rubisco. But in my opinion, I think that even increasing the chloroplast transformation rates, even just a couple hundredfold, you would make a drastic difference in the amount of Rubiscos that you could actually screen, the mutants that you could screen. And well, why is that the case? Well, why would nanotechnology be able to be used? Because, well, nanotechnology is based off of nanoparticles, and nanoparticles and nanomaterials are really everywhere already in the environment. So you have, this is something that I learned by going into this lab, is that the nanomaterials aren't this new thing that have just been created and because of new technologies. In reality, they actually come from the environment. So there are erosions, there's different types of sediments, there's different types of ways that these nanomaterials actually get into the environment without like any type of our fabrication. So, and already these nanoparticles are present in a lot of different products. So these cerium oxide nanoparticles and carbon nanotubes already have automotive, catalyst, electronics, coating, painting, all types of different types of uses. Because they create charged polarities that can be added to nonpolar surfaces, and it increases the amount of binding that you can have. And I mean, already there is a predicted carbon nanotube and cerium oxide particle concentration in the San Francisco Bay, for example. So it's actually like an interesting environmental toxicology question is, what types of materials and what types of cell biology properties would be needed in order for these things to be more or less uptaken into the organism. And there already is a mechanism that the proposed mechanism is for the uptake of single-walled carbon nanotubes into chloroplasts. So this is the dual membrane lipid membrane of a extracted chloroplast that is basically coated with the high polar carbon nanotube as it goes through the chloroplast. So there are mechanistic research done already in how these nanoparticles can get into biological systems. And basically, the research question was, if these biological organisms are already taking up these nanomaterials, then maybe we can facilitate and use these in order to deliver a technology. And how we do that is how we actually enable nanoparticles to become nanotechnology, and specifically chloroplast biotechnology applications. You start with a nanomaterial, so you have a carbon nanotube, carbon dot, nanorod, cerium oxide, mesoporous silica, and then you give these different properties and different coatings. You can attach different cargoes. You can have different size nanoparticles, and then they basically can have different charges. So these are all properties that you can kind of play with. And basically this functionalizes them for different purposes. So biomolecule delivery of DNA, you have chemical delivery that could be, used in those cargoes, or you could have an antioxidant capability with the cerium oxide nanoparticles, so like a scavenging capability. And and how would you get these in is the next question. So you have a nanomaterial that has a property now that's been functionalized for a purpose, but how do you get it into the organism? And specifically for a chloroplast, you would have to basically deliver it through diffusion, or you could do a foliar delivery with kind of like a spray, or more or you can do basically a needleless syringe. So without a syringe, the needle would just be pushed into the cuticle of the plant, and it would go through the outer algae or leaf cuticle, and then it would have to go through the plant cell wall, through the cytosol, right? So through the cytosol and around all of that. It would be coated basically. You'd get some coating of the proteins and stuff from the cytosol connecting to the nanoparticle, and then it would finally have to go through the chloroplast membrane. So that's how it could get in the pathway. It would have to get to the chloroplast, but then there would be a lot of nanotechnology based applications for chloroplast biotechnology. So there's, like we talked about before, chloroplast transformations. You could improve photosynthetic screening, organelle specific delivery. It could be a chassis for RNA, DNA. And then just natively, you could actually just improve the plant itself. So you could increase photosynthetic rates. You could increase their stress tolerance by scavenging ROS. You could have a host of different other functions too for bioplastics, biopharmaceutical production from these delivered delivered particles. And then, how small are we talking about? What are we actually seeing if we were going to see? So this is all to scale. So if you look at the glass bead on the left, which is 400 microns, that is the size of a normal. That's the size of the particle bombardment chamber that we were talking about before. Or no, sorry, excuse me. The gold microcarrier, which is on the bottom subset, that is the standard microcarrier of DNA that would be coded to it. The glass bead is used in transformations of algae, but in normal particle bombardment, you would have the gold microcarrier. And then basically, these are different types of nanoparticles and nanotechnology. Used in this research, though, the carbon nanotube. So we used a 5 nanometer by 100 nanometer long length carbon nanotube. And you can see that when compared to the tobacco mosaic virus, it's actually pretty much on the size of the tobacco mosaic virus. And basically, research has shown through analyses and through modeling that the higher charged nanoparticles do best. So in order to get into the cell and to get into the and to latch into, be isolated, to bind into the chlorophyll of the chloroplast, which is itself charged, right? You need a highly charged particle to get into there. So if you look at the top right there, that's a carbon nanotube with a chitosan, and then on the right here you have a plasmid DNA bound to a single-walled carbon nanotube. So this doesn't have a delivery mechanism because it's DNA that's just bound to a carbon nanotube, but this just is the modeling of where it can go based off of that. Previous research to ours did show that plasmid DNA can be delivered to plant nuclei. So this is GFP, intracellular GFP, that is being expressed by nuclei of the cell, that it was delivered by a PEI-coated carbon nanotube. So this is a carbon nanotube that was coated with polyethylenamine, and then there was and then there was dye bound to the DNA, and then that was shown here. So it basically was able to, it wasn't expressed within the within the cell, but it was actually using colocalization analysis to make sure that it was bound. Here, this is the actual GFP expression within plant chloroplasts. So this is within land plants. So needleless syringe delivery of plasmid DNA-coated carbon nanotubes, and then a fluorescent protein reporter that was then visualized through confocal microscopy. So this was basically the land plant version of what we did. You can have foliar delivery of cerium oxide nanoparticles as well. And it happens in a similar fashion, where basically you want high zeta potential, which is the charge for nanotechnology, and then that gets you have more functionalization that way. this is an ability for cerium oxide nanoparticles to be used and actually used as a chemical cargo boat, and to actually deliver based off of a targeting peptide to the chloroplast specifically. So that's basically just shows the functionalization, the capability of these nanoparticles once they've become nanotechnology. And through the basis of my PhD research hypothesis was basically there's huge advantages of nanotechnology over standard protocols, and you have the modularity that you need in order to adapt these new technologies for specific usages, and they can also be capable of in situ delivery, so delivering these biomolecules where the cells are, how they are. And then there's also unique characteristics that exist because they are on the nanoscale that we have really never seen in any other thing. In addition to my PhD research, we did, I did find that reproducibility was so important, and I did find that I wanted to give back kind of in a different, unique way through a journal of visual experiments. So basically, we took these cerium oxide particles and we used their unique ability to scavenge or de-stress the plant in response to stress. And we did a unique we did the experiment all on video. So it was just an interesting way that I learned how important reproducibility was, and I do think that especially in a biotechnology and a biotech capability, like the reproducibility matters so much, and because you're doing something 1,000 times, you're not just doing something for a figure. And I think that doing and utilizing unique ways to increase reproducibility in the work is very important. But my full research, my published paper was the DNA delivery by high spec ratio nanomaterials to algae chloroplast. So before this had happened in land plants before, but this was the first time anyone had ever delivered DNA to the chloroplasts of algae through nanotechnology. And we utilized single-walled carbon nanotubes. So carbon nanotubes are a high aspect nanomaterial. So that means that they are really, really skinny and really, really long. So if you imagine kind of like a buckyball here, or just basically a tube, a paper towel tube, it's just really, really long. But inside they're a lot less rigid than I imagined. And through AFM and SCM, you can kind of see that they're kind of like a hair. They can move a lot they're not as rigid as I would have thought. And these are, again, single-walled carbon nanotubes. You can also have multi-walled carbon nanotubes, but these are single-walled. And our main research hypothesis was that the lipid exchange uptake mechanism that we had seen in the isolated chloroplasts would also exist for the algae, since there's a similar cell membrane, and that we have because we were working with Chlamydomonas, we had not access to cell wall knockout lines. So the cell wall knockouts would have similar properties to basically plant protoplasts in our opinions. So and the high aspect ratio nanoparticle, so the single-walled carbon nanotube, would enter easier than something like a buckyball or something like that. So how did we create these? This was about 2 years of chemical engineering and a lot of trial and error, but we were able to reproducibly and efficiently create this process. So the functionalization of these carbon nanotubes with the polyethylenamine bound, and then basically through an electrostatic interaction. So without the PEI coating, the DNA and the nucleotide formation biomolecule, it's actually able to, the DNA is actually able to detach and then be transcribed or be used in this way. So we were, this was a functionalization for DNA delivery. And In order to create these PEI carbon nanotubes, we did different weights, so different lengths of the amino chain, so 10K and 10K, 25K, which is the molecular weight, which gave us different sizes and charges. So it was able to test our original hypothesis, and then we were able to bind the DNA to the carbon nanotube, and which is with just a 30-minute room temperature reaction, and then we were able to apply it to liquid cultures. So we did in situ, no mechanical force, so no type of particle bombardment, anything like that. So this was just in situ delivery of the liquid culture. So the functionalized dye DNA PEI carbon nanotube would then insert into the algae and go through the mechanisms that we were talking about before to co-localize into the chloroplast. And then to analyze it, we used confocal microscopy with co-localization analysis, and then did a myriad of different assays. So FDA, ROS, carotenoid, biocompatibility assays to assess that. This was the biggest part of the whole thing, was really creating and maintaining the same carbon nanotubes specifically, so that we were able to get reproducible results. So in order to do that, and how these interacted with the DNA was crucial. So we had to make sure that the DNA, with the right amount of DNA, was bound, because with too much DNA, it would clump, and with too much DNA, it would lose its charge. So how much do we actually load? So we tried different ratios, but the 1-to-1 was the one that seemed to work the best for the most applications, and the length, as you can see, is pretty varied. So it's really interesting how the length, it can be varying or between 200 to 500 nanometers in length, and these are 5 nanometer carbon nanotubes, by the way. So, but on average around a 600 to 700. And then now the 1-to-1 ratio of the DNA to PEI carbon nanotubes, this was the crucial thing of the charge, right? Before, it was the high charge carbon nanotubes that would be, co-localize the best. So, did they do well, and were they able to actually keep that charge? And yeah, so thankfully, we needed the most amount of DNA possible so that we could have the most amount of dye, so that we could visualize it, but we didn't want it to clump or lose its charge. And it looked like the 1-to-1 ratio was the best. And this is what it actually looks like. So this is the green here is chloroplast, and the magenta here is the dye. And the white spots are co-localized dye and DNA together into these merged images. So this is a confocal microscopy, and again, so this is the cell wall knockout algae. So this is the cell wall knockout, and as you can see, they're together here, and this is the full wild type cell wall. So it's less. You can see that there's less white. So there's less in the zoom image. There's just less events that happen, but also it was harder to get those results in the cell wall knockout, because basically the higher charged nanoparticles meant more co-localization, so it also meant more cell death because of that. So we got more co-localization, statistically significant, between the cell wall knockout versus the wild type. And the PEI 25K which is higher charged, had more colocalization than the PEI 10K, which is as we hypothesized. And this is kind of a population-based assay here, where we're looking at just a larger screen here. And as you can see in the top right, it's a lot. It's widespread. It's very there's a lot of interactions that are going around. These 5 nanometers with a 10 micrometer diameter organism, if you think about that in terms of a 6 foot tall person, these carbon nanotubes are the width of a 5 millimeter pencil lead. So these are like very, very small, and there's lots of them all constantly around them. So, and this is just for a control. If you're wondering, dye DNA without the carbon nanotubes, there is none. No signal. The dye DNA is not interacting with it, so it definitely is the PEI carbon nanotubes that are the carriers. And we saw very quick uptake of these things. So we're talking within an hour of the incubation, we were seeing co-localization of these dyed PEI carbon nanotubes, and specifically significantly more with the PEI 25Ks in both the wild type and the cell wall knockout. And then we also saw a dose-dependent response with transient reactive oxygen species. So, basically, the more reactive oxygen species there is, the more stress that they have. And we saw the increasing concentrations did yield an increase of reactive oxygen species, but then over time, that reactive oxygen species did wane. So that is good to see. And the cell wall knockout did show the highest highest amount of ROS generation. So presumably, especially at the higher rates, because there were so much cell lysine. And so then the question would be basically, what would be the concentration that would be the most biocompatible? So, dead cells with no FDA, which FDA is metabolized within living cells. So if there is none, there is no fluorescence. So we saw that the 30 femtograms per cell was about the best to actually get and create localization without calling mass cell death. And then we tested basically how lethal are these over 4 days. So we did, and we saw that the cell wall, the success of the wild type cell wall was capable of withstanding these 30 femtograms per cell rates for 4 days. And the 300s were good, but it did show decrease growth, and then 3000 would just die. So in conclusion, basically the PEI 25K and 10K were successfully synthesized, characterized, and with varying charges and sizes that we could finally test, and we had and produced a reproducible method in order to do that. The higher charge PEI 25Ks co-localized within the chloroplast at much higher rates because the higher charge, our original hypothesis was correct, and which is interesting because basically you don't need to have a cell wall knockout for chloroplast DNA uptake, which is something that isn't necessarily, some transformations do require an enzymatic digest of the cell wall and before you can transform DNA. So this was actually an interesting finding that we have. So it would be possible to transform with carbon nanotubes without having to do that enzymatic digest. And we did find that biocompatibility existed after 4 days. So 30 femtograms per cell was a concentration that was usable. And kind of like next steps, I mean, this was, pioneering work in a new field. So, how and where do I kind of see this happening? I think that there's, using ROS and FDA are good high throughput population-based ways of basically looking at different types of sizes and charges that would be good for uptake. And I do think that you could deliver reporter proteins that would be, expressed inside the chloroplast or, hopefully, plasmid to eventually try the transformations. We did We did try the transformations multiple times for over a year, actually, and I couldn't get it to work, unfortunately, with the pandemic happening. It was really excessively hard, but I do think that it definitely is possible. Um, the in my hypothesis, what would be the transformation that would actually work would be something with high charge, obviously. But the greater the width, the more likely it would be to clump. So it would probably you have to look at different types of ways of expressing that DNA, I think. And I think that just a simple vortex with sucrose recovery, would be all that you'd need. I don't think that you'd have to use particle bombardment based off of our research. Um, and then I think that a interesting future direction with this new transformation that could be organelle specific. I do think that there's really interesting dual antibiotic resistance selections that you could have with spectinomycin. You know, basically you could transform the nucleus and the chloroplast at the same time. I do think that would be possible, but the rate limiting step would be the chloroplast transformation. And additionally, I do think that in Chlamydomonas, it's a unique opportunity to actually look at mitochondria as a possible transformation organelle. It's only Chlamydomonas is only one of two organisms that's ever been transformed inside the mitochondria. So it as a research opportunity, it's completely unexplored to genome engineer the mitochondria for increased cellular function, and theoretically, it has the same type of evolutionary trapped mechanism that exists for mitochondria as do the chloroplast, and that once they were engulfed and taken into the cell as an organelle, they stopped evolving the genome for increased function itself. The idea was that it's evolving in order to become and losing its DNA and exporting it into the nucleus. But could the mitochondria itself be improved through genome engineering? And I think that it is interesting to think about. And also, I think that this is like an interesting possibility for in situ delivery of unculturable microbe communities. So the ability for microbiologists in order to work with a microbe inherently relies on our ability to transform it. And transforming it requires us to culture it, right? Because you have to grow that grow that organism after you've inserted the piece of DNA. So inherent to synthetic biology and the needs for microbiology is an in situ delivery platform that you could actually create a way for these microbial communities to be monitored in situ while these microbial communities exist. Because when you culture one of them or monoculture one of them, you're destroying that microbial community, and it's hard to understand how it exists. But with synthetic biology, I do think that a nanotechnology delivery platform could enable that research. So then after my PhD, I went to BASF, where I was where they needed to create Pichia as a host development platform for bioservices to create different types of antibodies. And I basically, I would research potential targets for knockouts. I would do different types of research into genome engineering targets, and basically I would be designing doing the vector construction, the molecular cloning of the DNA pieces together, doing the actual transformations, then doing the screening, the sequencing, and then handing off to the fermentation team, where I worked with one other person on the team, a scientist under me, and we worked through it together. Uh, Basically, I was doing genome engineering for of these hosts with through knockouts, targeted inserts through homologous recombinations for eventual protein production, and we had some wins. We developed the and used a molecular biology knockout tool that was off patent because they wanted to do these mutations and this engineering without CRISPR because they wanted something that they didn't have to pay the royalties and the licensing fees of, since were the parental strains that they were going to be using for a good time. So they wanted to be able to do it without the licensing fees. So we created over 80 strains in 18 months. These would be knockouts and along with targeted inserts, different types of inserts, different types of chaperones that we were looking at. And then also a huge win was our ability to increase the wild type strain homologous recombination rate. So I through an altered transformation or through an altered competent cell protocol, we were able to increase the rate of homologous recombination from 1.5 to 5, which was a huge benefit for us in order to make those strains. And then I also, as I was leaving, we were working on developing a high throughput protocol to generate over 30 strains at once. That was my final project that I did. And I think that honestly, this was like the proof of concept experiment that generated 30. I think that we could, I was planning on doing a whole plate. So I think that you could easily do 96 strains at once with this protocol. And we used a lot of automation to do it. So it was very nice. I mean, to be able to use the colony picker and automated liquid handler again. So, going back to the liquid handler on the Hamilton was pretty awesome. And, through the automation platform, basically we were able to automate the entire workflow. So from transformation to the screening to the sequencing, everything was run through robots in order to make it in about 2 weeks. So it was very fast. And then, through that, and through these strains that we did, we were able to increase the antibody production of a target by 20-fold. So it was incredible. It was an incredible feat, and we had. We were well beyond the target amount, and very happy in order to do that. And in general, just in closing, I think that microbes are just the final frontier. I think there's such huge opportunities in the world of microbial engineering. High throughput strain engineering specifically has the ability and access to large synthetic mutant libraries now that we have huge combinations of different types of proteins that we can produce now because of it. We have automated liquid handlers that really take care of all the physical body bottlenecks that would be needed so that you can do a lot of this stuff robotically now. And then in order to analyze and create these strains and to confirm them, next generation sequencing allows these mutant libraries to be screened through direct evolution approaches. So you can use the ability that is native to biology and using evolution as an approach in order to screen these large mutant libraries. It's like never before. And I think that it just allows so many opportunities. And then on top of that, there are utilizing new AI-based like deep learning approaches to basically crunch this phenotypic assays and allow researchers now to see what are the things that we're missing here. Like what are the correlative variables that are really going to be able to increase the protein production. that we might not be noticing. So just throwing tons of data into these phenotypic assays and getting a lot of data that can be pushed into deep learning. And Also, I mean, as research goes forward into microbes and the breadth of microbial diversity that's out there, you're just going to have more and more diverse chemical products from, from food to materials to fuels that will be able to leverage a lot of these new engineering approaches, because through fermentation you have exact precision, and through that you can really fine tune the process, and very crucially with no externality like variables. So as opposed to creating lipids or plant matter, for example, through switchgrass. You have, which has a lot of climate variables and runoff problems and all that kind of stuff. Using fermentation as an ability in order to create biomass is definitely the future that I feel is going to be what we utilize as humans in order to solve these renewable and sustainability problems that we have. So I'm just very excited about the future of microbiology, but I am a little biased as a microbiology PhD. But thank you so much. Any questions? Okay, so 50 minutes. Okay, let's see here. I have some really straight away things I need to go through. I should have just done it while I was doing it. I knew there was going to be some stuff. Okay. Hello. How was that? What's up, guys? Oh my gosh, there's people here? That was weird. Talking to myself. How are you? Good evening. What do you guys think? Was that good? What do you think? We're here. All right. Oh, those are the amount of vaccines that we've gotten as a community, because we love vaccinated people here. Cool, man. Cool. I like it. Oh, thank you, Smooth. Thank you. Yeah, Rapture. I agree. I think like going through it, I don't normally I'm staring at it, so it is a little weird. Like I don't normally read as much. I understand the um uh okay, I don't know if we could okay. What's up, dude?
Hey, how you doing?
Good.
The presentation sounded really great.
Okay.
I everything you do, trust me, I enjoy a lot.
Okay.
I've been following you for a little bit.
Cool.
The alms, I'm telling you from a I'm a blue collar worker, maintenance guy.
Uh-huh.
Uh, I lost count at like 300 alms before you actually need to pause, just
Okay.
to so you can get yourself straight. I know it's can be stressful shit for a job interview and crap. I interview maintenance guys all the time, maintenance man, but the ums all the time.
Okay.
That's usually going to be a red. Just saying, this can usually be a red flag on some potential people. You know what I mean?
Sure.
Uh, just positive feedback. That's all it is. But the presentation was freaking awesome. But the ums, I think I lost kind of like 300 and something, somewhere like half hour, 45 minutes into it. But I watched the entire thing.
Cool.
I
Thank you.
love scientists. Trust me.
Cool. Thanks, man. I appreciate it.
That's yeah. It's just giving you a heads up. That's ums and ands. Most people don't like that shit.
Gotcha.
Oh shit, my bad.
It's okay.
just a little positive feedback for you of what's, like I said, for my job skills, which is nothing close to yours, but that's what 9% of the people look at is that thing. It's
Gotcha.
just
Okay.
take a pause and don't say anything. Just pause. And I know it's stressful changing jobs. I know I do it all the time, every 5 to 8 years.
Gotcha. Awesome. Thanks, man. I
All right,
appreciate the feedback. All right. Have a good one.
you
All right. Thanks, man. Thank you for that. I appreciate you, Alex. All right. Did I get it right that you could use wild type algae in the second? In the second? Yeah, yeah, you could. Yeah, yeah, that's good feedback. I didn't realize that I was doing it so much, but okay, cool. Thank you. When you're interviewing for a scientist job, you're expected to give an hour-long presentation on your background research. Hi, Richard. How's it going?
Hey, Dr. Greg. We interacted a couple days ago.
Uh
Got to
huh.
tell you, um, I really did appreciate. Um, I appreciate a different side. More to the point, I really did appreciate you sending me links to some of the stuff that I had questioned you about.
Okay.
Um, let's say this: Alex needs to realize that he said that he's a blue collar guy. He
Yeah.
just has a different level of experience. Um, and he has a different level of experience, and he needs to be, you know, and be very positive about that. But anyway, I'll tell you what, man. I enjoy what you do.
Well,
I
thank you.
truly appreciate everything you bring up. And more to the point, um, it's just it's just really positive. I mean, primarily open and frank conversation is what we as a species need.
Mm-hmm. I agree.
Well, I'll tell you what. Let me ask you this: in your, what is what I'll ask you this: what is your? I mean, I know it's not necessarily
Mm-hmm.
your area of expertise, but what is your thought process or what is your opinion of Eric Weinstein?
He's just a fraud, man. I mean, is he the older one? Brett Weinstein,
I
the
think he's the older
biologist,
one.
is his PhD is a
Well,
joke,
Eric
though.
was
It's
the
crazy.
physicist.
Yeah, I mean, I'm more familiar with his brother. I've actually read his dissertation. It's a complete joke, man. He used other people's research data and he never published it. He did a 10-year PhD, and they were obviously just like they just pushed him out the door, and he was
And you know
he's
what?
yeah, it's a
I
joke.
appreciate that because I've seen him show up on certain things, and it's like this guy is trying to this guy's trying to sell something that
Hmm.
I don't think is necessarily correct. But I wanted someone that actually is in the field to actually just talk about it. I
Yeah.
mean, if the guy is a fraud, or is, let's say this, maybe not a fraud, but if someone is not competent in what they do,
Uh-huh.
then you know what? That's all I need to hear. I appreciate that.
Yeah, he's an evolutionary biologist, and it was basically a modeling study that he did, and I mean, it's not impressive at all. Like as an academic, he didn't really do any actual novel. Um, he didn't collect any data himself, and he didn't like his figures are laughable. I mean, he
And you
didn't
know what?
really do anything that was PhD worthy in my opinion. It blows my mind that he got
I'll
one
tell
actually.
I'll tell you what, that is incredibly fair. Then let me ask you about there's one other person that has kind of come across my come across certain things that I've looked at. Um, what is your take on God? Um, okay, I'm having a brain skid. I apologize.
Hmm.
Um, I mean, first of all, and we had part of this conversation the other night. I believe in vaccines.
Yeah.
Um, I believe in. I do indeed trust the science. Um, but and one of the things I commented upon the other night was I said I do believe that we are on the. I do believe that we are on the precipice of kind of a paradigm shift. Do you remember that?
Yeah.
Well, the reason I say that, because I mean, it was like dark 30 when I made that comment.
Mm.
But the reason I believe that beyond that is because I think where we are right now, from everything as a non-scientist, I must say that initially, but I really do kind of believe that we are sort of almost at a. I believe that we are really sort of on the precipice of saying, you know what, we need to expand our view.
Yeah.
I mean, because there are certain things that science does indeed answer,
Hmm.
but there are a lot of things that our current understanding of what science is does not and cannot answer,
Okay.
in my opinion.
Sure.
And you know what? As a result of that, I certainly want your insight because, as I told you in a DM,
Hmm.
um, I do kind of respect. I do respect dissent, and I want people to dissent. Um, because frankly, and I got a lot of hate for this, um, only through dissent do we come to the truth.
Okay. I mean I don't think that's I don't think that I would agree. I mean, through consensus is how we get
No,
the truth,
okay,
and like the dissent is part of a process, but you have to contribute meaningfully. The problem with just dissent is that it's reactionary. So you're just reacting to someone and say or something and saying no, this isn't how it is, and it's like, well, you have to show how it is. This is Karl Popper, right? This is falsificationism. If you aren't if you aren't creating an idea that can be tested, then you are not adding anything to the conversation.
and you know what? That is a very fair point. And but what I mean by that is what I mean by dissent.
Uh-huh.
And let me be very clear on this point. Um, I want really smart people who are actually who are in this process to actually maybe disagree. And
We do though.
disagreement, in my opinion, actually leads us to thinking differently, maybe taking a different sort of mindset, a different perspective, and saying, you know what, maybe there's some value to that. Does that make sense?
I um it, okay, so the process that science works with isn't what we're talking about right here, where it's not like this conversations like this. The process
Okay, I
that which
apologize
no
if I
no
brought that in.
no
My
no
my
no no
apologies.
that's fine. I mean, no, no, there is a mechanism that their dissent is capable and it is through the scientific process, and that's how you battle that. It's like, I do jujitsu. Okay, so jujitsu like has belts and gyms that you go through,
Sure,
but then there
sure.
are championships that where people have the tournament and then they figure out who the best is. And then there's, someone that is the world champion, and that's who the world champion is. But these people are, also training, and if I somehow tap the guy that is the world champion, who's the world champion? It's not me. It's the guy that did the tournament, and he is the world champion, right? So it's in the
And
same
that
way,
is very fair.
and it's the same way that
That
like
is very
you
fair.
you can say, hey, this thing sucks. I don't like how the methods were done. This wasn't the correct thing, and okay, prove it. You have to do the work. The tournament in this case is the publication, and you don't get to kind of shortcut this whole thing and say I'm the world champion because I tapped you. Just like you get to say, oh, I don't get your entire study is invalidated because I have a little quip of it. That's just not how it works.
I understand that and I appreciate that. And you know what? Of course, you're correct.
Uh huh.
Um, but I guess the only thing I can say beyond all of it is I just think that we do need, on some very fundamental level, and this is something that I've seen, and it's kind of played out for a while, um, that dissent is kind of shut down, even though the dissenter was inevitably proven correct.
Yeah, I don't think that that's true, man. I don't think every single conspiracy theory
No, it's not a
was out.
conspiracy
No, no,
theory.
no. I mean,
It's not.
no, no.
Please
For
go
real,
ahead.
everyone that had an opinion about COVID was able to say what they wanted. Nobody was silenced. Like everything was tested. Hydroxychloroquine, ivermectin, like everything like that. People were
And
able
I'm
to
not even
to
going into that. But
Okay,
you're absolutely correct because, like I said, you sent me a link
yeah.
to some of the stuff the other day, and I'm still reading through it. Um, and a very, I look forward to having a conversation with you regarding
Hmm.
this, um, because maybe you can, because of your intellect, your training, can
Hmm.
actually tell me where I'm wrong.
Hmm.
But there are, all I can say is this. There are. I look at the guy that actually won the Nobel Prize for proving that ulcers were literally uh, caused by, you know, H. pylori.
Hmm.
Because a lot of, and I'm not trying to be a conspiracy theorist, but this is one of those instances where I sit there and I say, there is a consensus, but maybe someone with a different idea might be right. So, we need to look at that. And by the way, here was a guy that actually said, okay, well, you're saying I'm not correct, but I'm going to drink a glass full of this bacteria. I'm going to have a horrible bleeding ulcer. I'm going to take an antibiotic, and hey, surprise, surprise,
Uh-huh.
I'm cured.
Okay. I mean, again, it's just anecdotal, man. Like that's not how it works.
It's not anecdotal. Um,
Yeah,
I
just
mean,
one thing. Yeah, just one study. I mean, you have to look at the mechanistic. You have to look at the mechanism, how it's working. You have to prove it through studies. You can't just like circumvent the process just because of one person, that might not be the reason why that's happened.
I appreciate that. Um, And look, I certainly understand, and more than anything, I certainly respect. By the way, Kathleen, I'm not going to lose my shit. I promise you. But all I'm saying beyond all of it ultimately is this. More than anything, I appreciate you taking your time, chatting with me. Frankly, you are probably one of my favorite scientists out there.
Thank you, man. Thank you.
And the reason for that ultimately is this. One, you actually do educate, because that is what science needs to do. I would like to say that, I mean, science needs to say, okay, we're doing this because this is where we want to go. More to the point, these are the facts that we have actually figured out. And this is why, all I'll say is this. It always comes down to this. Make your case. Make your case strongly.
I appreciate people. Scientists will always want to be held like accountable for things, because there's a good faith self-reflection that happens within the scientific process, where you try to make things better and you try to see what actually happened, and I think that people exploit that, because there's like a hyper it's like a hyper accountability for science, but then for like the conspiracy theorists, we're going back to COVID just because I guess we always do, but the conspiracy theorists and stuff are never held accountable for anything that they have. Any type of mass death that's happens because of the vaccine, that was wrong. Hydroxychloroquine wasn't good. Ivermectin wasn't good. But then you have all these people that peddle that stuff constantly, and then now they're able to say at the end of COVID, after all these people were lived because of the vaccines, are able to say, oh, actually, it's like science was wrong the entire time. And it's like, what? Like, the science was right the entire time, actually. It was the conspiracy theorists that never got anything right the entire time.
And you know what? That is a very fair point, and I appreciate that. By the way, I know you've got a couple other people that are waiting. Thank you for actually listening to me. Again, I am reading the links that you sent to me.
Cool.
I look forward to having a conversation with you regarding that.
Okay. Sounds good.
Okay. Sounds good, man.
Tell you what, man, you have a great night. Thank you so much.
Thank you. Appreciate it. Okay. Okay, making money. Okay, guys, I'm going to take off in 15 minutes. So thank you for everyone being concerned about me going to bed at the right time. Thank you. Making money with Sandra. How's it going?
Hi, how are you?
Good.
I have a question. I had the flu shot,
Mm-hmm.
about 15 years ago, I should say, or 20. And after that flu shot, a week after, I got really sick with the flu. I was nauseous. I had the runs. I went to the hospital because I was shaking and so nauseous that none of the medicines to get rid of the nausea was working. So I went to the hospital. They gave me a shot twice. Nothing worked. They said, take this medicine and you can take it home. But they said, don't take it right away. I was like, you know what? I am still really nauseous. I'm going to take that medicine when I get home. And I had it for about a whole month. I was so sick and so weak. I think I might have been allergic to the shot. So after that, I thought, you know what? Forget the flu shot. I'm going to let my body, because I'm young, let my body just get immune to it all. Why? Because I feel that if you take all these shots, your body is not going to build up its own immunity, because it's going to get used to all these vaccinations, and it's going to require your body to, I can see when you're kids, you do need vaccinations. Yeah, there are
Okay.
vaccinations you do need, but the flu shot, I just thought I would let it build up my own immunity because I'm still young and healthy, right? But why
Okay.
was it? Do you know?
I mean, no, I can't tell you 15 years ago why what happened, but I mean, the flu specifically this year is really deadly for kiddos, so it's really important that everyone gets the flu vaccine this year. So, I mean, even the CDC, even RFK is recommending that the flu shot, that you take it for anyone 6 months and above. So,
I'm just scared because I don't want the same reaction to happen. I have that fear. It felt like I was on my deathbed because after I got that, like, even my mom had to come over and help me, and I was so weak and I couldn't work. I couldn't do anything. I just was throwing up constantly.
yeah, if you actually got an actual flu infection, it's awful. I mean, I've only gotten a couple times in my life, and it's been, it was off. Like, it floored me for like a week, and I was like, wow, I understand why people actually die from this. It's crazy.
But I had it for a whole month. That's the thing. Like that's why it was so awful. Like
Dang, I'm sorry. Yeah, yeah. I've gotten the flu shot every year of my entire life, and I've been okay. So,
I've got the vaccine. I got that a couple times, like 3 times, but I won't get it. I'm afraid to get more vaccines because I heard of people actually getting the COVID vaccine shots and then they get ill
no,
from it.
no. I mean, you don't get ill. You get vaccines. Like, basically your body creates the antigen, and then your body responds as if there was an infection, but you're not actually getting sick when you get a vaccine. So, people think that it's an adverse side effect. It's not. It's your body responding to the antigens it's an immune response, and part of that immune response is an elevated body temperature.
Yeah, okay.
Hmm.
All right, that's all I have to say. But you're doing great. I love what you do
Thank you.
and everything. And it's good science because actually my mom was at first afraid to get her vaccine
Hmm.
shot. And my brother, he's like, oh, they've been studying science and all that for years. So
Nice.
they already had the vaccine pretty much
Hmm.
because they've been doing it for so many years. So he's
Okay.
like, Mom, you got to get the vaccine. So she
Good.
got it because he talked her
It's a
into
good
it
kiddo
because
right there.
you know. All right, bye.
All right. Thank you so much. Thank you.
You're
Thank you,
welcome.
Sandra. Have a good one.
Thank you.
All right. NB. What's up, NB? How's
Hey,
it going?
I'm doing okay. How are you?
Good.
Good. I'm gonna try to choose my words carefully, but I'm having a lot of like brain fog tonight. So if I say the wrong thing, chat, don't come for me. Um,
I'm sorry to hear that.
yeah. So you know my story with my benzo-induced like neurological dysfunction and what I'm going through with my withdrawals with my benzodiazepines.
Sure.
And I got vaccinated, I got my MMR, I got my COVID, and I got my flu.
Hmm.
I have two different things happening at the moment that I think might be happening to me. I think I might either have like something called serotonin activation syndrome,
Hmm.
and I just haven't noticed it, or I think that my immune response to the vaccines has shaken up my nervous system a little bit somehow. Someone told me that vaccines have not been proven to do anything with our neuropathy, but I looked on Google Scholar and there is quite a few studies to show that it does, but there's not a lot of sightings about those studies. So I was just curious if you knew anything about, and I'm with you, by the way. I know you're going to say that if I got the actual diseases or viruses, that it would be worse. And I
Right.
agree. So I'm glad that I got vaccinated, right? Because obviously if the vaccines made me feel this way through my bind, then and made my symptoms exacerbated, then the viruses would have made it 1,000 times worse, right? But
Yeah.
I'm just curious if there's anything to do with vaccines and viruses and how they work within neuropathy, and what you know about that.
I don't. I mean, directly I don't know, but I would really focus, especially since you got the MMR one, and that's been known to kind of like put people on their butt a little bit. I would focus on not um, spending so much mental health like on this, and try to just take walks, sleep more, drink water. I mean, not to downplay with what you're going through. And I understand that you're going through something, but I feel like just constantly reading about it, constantly thinking about it can kind of like could it hasn't been that long, so I would just like just keep try to be a little bit more active, try to not think about it. If it progresses to the point where you know there's extreme pain or something like that, then I would obviously get it. Like then I would start being concerned. But I'm not a physician. I can't tell you like medical advice. But I would think that just constantly thinking about this stuff and being in your head can be, it can be a bad place no matter where or what is happening to you. You know what I mean?
Yeah, it's interesting because I was trying to like, how do I say, like, almost disassociate myself from my symptoms, and then I
Mm-hmm.
just got a new doctor, and she asked me to log every time that I have a symptom, so that way, like as a physician, she's able to have these things, and so now I have a shortcut with my Siri, and so now every time I have a symptom like tingling or nerve pain or like a
Mm-hmm.
headache or a backache, I say, "Hey Siri, log a symptom," and then I log it, right? I'm not diagnosing myself. I have a diagnosis of mind, and then I was
Mm-hmm.
researching things so that I could bring it to my doctor because doctors appreciate that,
Yeah.
Robert. Anyways, so that's all. But so I am bringing this to my doctor on Tuesday. I just didn't know if you knew anything about it,
Mm-hmm.
and it could be either or, right? Or it could be a combination of everything that I have going on. I will say that I do feel a little bit better, and I'm starting to feel better, and I am doing this. Like I went on a nature walk.
Good.
I'll send you the pictures on Discord in the PNW, and it was the most beautiful, like
Nice.
magical fairy forest I've ever been to. So like I am starting to be more capable of doing things, but I did notice a significant increase in my neurological symptoms, which I found to be a little strange. But I do see what you're saying. I will say I do get a little psychosomatic, but I don't think that's what's happening here.
Mm-hmm.
I think what's happening here is that my doctor did ask me to just start logging my symptoms, and
Yeah.
so I,
In
I
good
think
faith.
just
Yeah.
noticing them more, you know?
Exactly. Yeah. I think that's totally fine. I mean, that's good. I mean, I would just do what you can in terms of keeping your hopes high, and keeping your energy depleted from physical activity and stretching, and that's always like I love a good stretch. I've been stretching more and more, and yeah, like your walks. That's a great idea. Hikes every day. Just doing focusing on that stuff is gonna it's gonna get your mind off of it, and especially since you got the MMR, and there is a a little bit longer of a reaction to the immune response is a little bit more than normal vaccines. So that probably is what it is.
Yeah, I hope so. And just really quick debate, Doc. I didn't say serotonin syndrome. I said serotonin activation syndrome,
Mm-hmm.
which has actually been studied to, like, I have a higher, because I'm withdrawing from benzos, I have a higher chance of getting serotonin activation syndrome simply from starting a SSRI, which I did recently.
Mm-hmm.
So there's a very high chance that I do have serotonin activation syndrome, and have had it and just don't know. Debate, Doc.
Mm-hmm.
So that's something that I'm going to be bringing up to my doctor, and I found studies on it and on Google Scholar, and I was able to send them to her via our portal.
Nice.
Gotcha.
Right on, man.
I just want to clarify it super quickly. Um, so I'm not exactly sure what serotonin activation syndrome is. So maybe I'm thinking of the wrong thing. but we did learn about serotonin syndrome in school. And from, what I've seen, I've only seen it like once, but from, what I saw and then from my kind of like understanding from school, it's a pretty serious condition that's characterized not so much by like, neuropathy or neuropathic symptoms, but a lot of like kind of autonomic dysfunction. So like sweating, tearing, diarrhea, other types of gastrointestinal distress, and then a lot of like muscle hyperexcitability. So like, difficulty kind of controlling muscle movements, clonus. So it's this phenomenon where like you have a reflex to something like, or reflexive action to something, and then your affected limb, like whatever you've had the reflex in, gets kind of caught in this like sort of constant reflex arc where you kind of keep twitching that same extremity over and over and over again. So it's a much kind of larger, more global, kind of more serious phenomenon, I think. So my guess would be that if you were actually having serotonin syndrome, you probably wouldn't be able to like be up here, kind of talking to Dr. Greg at the moment. So that's the only thing that I was kind of curious or questioning about. Not invalidating any of the other stuff you're going through by any means. That all sounds very challenging. I was just kind of questioning about the serotonin syndrome specifically.
Yeah, so I can send you the information on it. Like I was saying, serotonin syndrome and serotonin activation syndrome are two different diagnoses. But yeah, I can send you the information on it if you'd like to see it. So
Yeah, for sure.
serotonin activation syndrome happens when you're getting onto the medication, rather like when you're getting onto an SSRI or when you're tapering off of one, rather than when you have too much of serotonin flooding into your brain. It's kind of similar, but it's not the same thing. And so it causes things like increased agitation, anxiety, some of the same symptoms that you have with bind. And so it has a very increased likelihood of actually exacerbating my bind symptoms.
Gotcha.
Like akathisia, if you're familiar with the term akathisia, it has a lot of like akathisia type of vibe to it. So, but yeah, I can send you some info on it. I don't want to flood the rest of your live with this because I know you're getting off Dr. Greg, but I appreciate you coming up.
Yeah, for sure. No, I definitely appreciate that. Thank you.
All right, cool. Thanks, Debate Doc. Thanks for coming up, bro. I hope everything
thanks.
works out, Envy. To keep going. Don't worry. Okay, it's gonna be better. This stuff will fade hopefully, and it'll be soon.
Yeah, thank you so much, and I appreciate it. Yeah.
Yeah.
And I feel better already. So I'm
Good.
like tomorrow's gonna be a better day, and then the
Yeah.
next day is gonna be a better day, and hopefully I'll stay in this wave for a minute. So thank you.
Good. All right. Nice talking.
Have
See
a
ya.
good night. Sleep well. Bye.
Thank you. All right, guys. It's a fast one, a quick one. Thank you for everything. It was good times. Happy we did it. We did a quick stream today, but I gotta go to bed early. I gotta sleep. Thanks for running through my research background with you guys. I've seen you, Ness. Yeah. Shout outs. Oh yeah. Before we do that, thank you. Real quick, guys. Thank you guys so much. I appreciate you. Thank you so much. To the last super fan, thank you so much, Dak. Thank you so much. Thank you to the Patreons, Mrs. Dr. G, Ado, Call Me Ness. Thank you so much. Thank you to the top gifters, Kathleen, Ducray, Bailey, Kellen, Cher, Keisha, Jared. Thank you, Carlos guy. Thank you to the top likers. Thank you, Jared, Kathleen, Bailey, Helen, Cher, Keisha, Ducray. Thank you guys so much. Guys, my link tree, I'm posting things on Instagram now. I'm an Instagram guy. You guys are all 55-year-old and up women, apparently. You guys hang out on Instagram, so I'm a yeah. I'm there. Yeah. For sure. Call me Ness. We will. And yeah, well, there you go, Sandra. There you go. You are my target demographic. You are my target demographic, apparently. All right, guys. For real, though, thank you guys so much. I appreciate you. Thank you, Sharon. Thank you. I know I'm 38. Gonna be 38 forever. It's crazy. I know. Thank you, Debate Doc. Thank you, guys. It's gonna go good. I gotta add some slides. I gotta talk a little bit more. 50 minutes wasn't enough. Gotta add some stuff to it. So put some more stuff in there. Thank you for the walkthrough. I appreciate it. I'll watch my verbiage. Did you guys think I've used a lot of filler words? I think I'm more comfortable here. So maybe I just like turning my brain off, and I hadn't seen these slides and gone through this presentation a little bit, so I was using more filler words than normal. That's what I think. So yeah. Okay. I'll listen back. I think that's good feedback though. I do consciously try to not use filler words, so it's good to know if I was. Thank you, Carlos. Oh yeah, it's gonna be on my YouTube if you want to watch it. That's a good point, Bailey. I think that's a good point. And when I present, I am really, I don't know. When I present, especially in a formal setting, I make sure not to use filler words, so it's on my brain. But here, I'm just relaxed. I'm chilling, and maybe because I hadn't gone through the presentation before, I was using it more than I would. That's what I'm thinking. I don't know. We'll see. We'll see. I hope I get it. Peace. Have a good one. Bye, everyone. Bye. Thanks for coming. Thanks. See ya. Peace.